Okay, let's talk a little bit more about the control of genes. We've talked a bit already about the fact that genes have an anatomy to them, right? Genes have regulatory regions and they have coding regions, right? Coding regions of the gene are the exons, and they contain the information necessary to generate the primary structure of the protein. That's the way that the amino acids are stitched together during translation. Okay? The regulatory regions of the gene depend on whether we're talking about a prokaryotic or a eukaryotic context, right? In a prokaryotic context, gene regulation includes the operon, which can either have an inducible or a suppressible structure, right? And the promoter, okay? And we should also include the possibility of secondary structure in the message that can form depending on the rate of translation that can either inhibit or promote production of that particular gene product. Okay, and then finally, the amount of time that the message remains active. Okay, in the prokaryote. In the eukaryote, the regulatory regions include the promoter and the 3' and 5' untranslated region, and introns present within the structural component of the gene. And there is even evidence that there may be some regulatory components in the exon portion of certain genes. We should also point out the phenomenon of trans-splicing and of alternate splicing, and that can produce different messages from a single transcript, whereas in the prokaryote you can make a polycistronic message, meaning you can have more than one protein produced off of a single nascent transcript. In eukaryotes, transcription and translation are not coupled. Transcription happens in the nucleus, translation on the rough endoplasmic reticulum or in the cytoplasm, whereas in the prokaryote, transcription and translation are coupled. They happen at the same time. Okay? So gene regulation involves the turning on and off of genes, while expression has to do with the pattern of the production of those gene products. In other words, where are they made, okay, and when are they made, right? The control of gene expression lets cells produce different kinds of proteins when and where they're needed. Our earliest understanding of gene control came from E. coli, and we talked about the operons, right? We gave examples of suppressible and inducible operons in prokaryotes, okay? And we've talked about the process of development as well, right? We've noted, for instance, that when we're considering development, right, we have several processes that contribute to the production ultimately of the organism, right? We have mitosis, we have growth, we have morphogenetic movement, and then finally we have the one listed here, number 4, which is differentiation. Okay, and all of these are happening during development at the same time, and they all involve chemical cues primarily encountered outside the cell. So there's an interaction between receptor and ligand that guides gene expression. Okay, differentiation is the change in fate of a particular cell from any possible fate to many possible fates to a single fate. Okay, mitosis is simply cell division, growth and increase in size, and morphogenetic movement, the movement of whole cell populations from one location to another following chemical cues. Okay, so differentiation involves the regulation and the turning on and off of specific genes in order to guide a cell down a particular fate as a result of chemical cues encountered in the environment. Okay, most of the cells in the body of an organism, a eukaryote, are going to have the same DNA from front to back. The only exception would be the lymphocytes and the gametes. Okay, they will have differences in their DNA from cell to cell. Okay, the lymphocytes because of germ cell recombination, and the gametes because of crossing over during meiosis. Okay, the differences between cell types as a result of the difference in the use of the information, not so much the difference in the information in the cell nucleus. Okay, so one of the things we've talked about already is this idea of X inactivation, right? In females, there are two X chromosomes, right? But in order to produce the same gene dosage that you have in the male, right? Here's the female, and here's the male, right? We want to have one active X per cell. So in the female, what happens is that one of the X's becomes inactivated, becomes what's called a bar body, okay? And which X it is depends. It's a stochastic event, meaning it's random, so the X kind of crumples up and becomes heterochromatic. Heterochromatic. Okay? Meaning that its DNA isn't accessible for transcription, so it's not used. So every female is a mosaic. Okay? One or other X chromosome is active in the female. Okay? That's for most vertebrates. Okay? Insects do some funky things. Chickens do some funky things. Birds do some funky things. But for most, let's say most mammals, it's the XXXY sex determination. Okay. Tortoiseshell cats are found in females because of this idea of bar bodies and pigment genes being on the bar bodies, right? So we have the epistatic gene that's above all of this that controls whether or not pigment is produced, and then underneath that we have the either either black or the copper-colored fur, depending on which allele is active in which X chromosome. That's why calico cats are always female. Okay, female cat. Okay, variety of possible colors. You can see them distributed here. Okay, always females. Okay. When we look here in the embryonic stage, right, we got the orange and the black fur allele. Random X inactivation is going to yield one or other being active, and the descendants of that cell will have the same inactivated X. Okay. Genome-wide expression studies are possible as a result of microarrays, which we've talked about a little bit already. A microarray is simply a mechanism to hybridize the expression products in a cell under a particular set of conditions to DNA probes that are homologous to different gene families. These lie on generally a slide, and we take the messenger RNA from the cell under the particular conditions in question. We harvest that messenger RNA, we make cDNA from it, and we can make it fluorescent by incorporating nucleotides that contain dyes attached to them. We bind the cDNA to the microarray, we wash off the excess, and the complementary cDNA is detected by fluorescence on a slide. Okay, so you can see the basic layout here, right? And we've used these microarrays for medical research to identify chemotherapies that will be most effective against certain cancers, okay, so that we can personalize cancer treatment so that it's more likely to be effective against a particular type of cancer. Remember that we want to think of cancer as a symptom, not a disease. Okay. The disease is mutation in genes control the cell cycle, right? DNA microarrays may usher in an era where medical treatment is personalized to each patient, especially in the case of genetic disease. And microarrays can also reveal the profile of gene expression in an organism over its lifetime. So this is a look at how microarrays are used.